last week we talked about really the derivation of the science and how it's an umbrella science and all the disciplines that are underneath it. We talked about some of the individuals who brought on some of the changes necessary to move away from what was religious dogma to what is now scientific understanding. Um, and and then we talked a little bit about some of the good things that microorganisms can do for us, including bioremediation and CRISPR technology and all those kind of things. But we're going to continue on today with looking and moving away from the golden age, those kind of things that led to the foundations of the science, to the modern age. And then we'll talk about classification, and then we'll talk about tools. And that's probably all we're going to have time for today. And then on Thursday, we will start with talking about the cells. We'll talk about bacterial cells first. We'll start off with cell walls. We'll talk about plasma membranes and glycocalyces. And um, and then in lab today, we will cover protozoal parasites. And in lab on Thursday, we will cover um, metazoal parasites and exoparasites. So that next Tuesday, if we're online or in the classroom, we'll review for our first practical. And your first practical is next Thursday. Book it. It's ready to go. Okay? All right. So, how do we defend against these infectious diseases? Well, we try to collect data. And we collect data from the community. I don't know yet, Sarah. I'm still kind of far away. Maybe I'll know by the end of this day. But we collect data, and we collect data from the community, but we also collect data from the individual, right? And so if you think about that, there's serological data that we collect, right? Blood work, things like that. There's immunological data that we collect, right? And then also we have chemotherapeutic drugs that we can utilize, right? So in some cases, these antibiotics work just fine. However, I will tell you that we are now in a place where there is so much drug resistance, right? That we need to address that in just a second. But so microbiology, the future looks good, right? I'm a microbiologist, so maybe I'm a little bit optimistic because I'm biased. But for every one answer that we have to the questions we ask, we always have more answers that are derived from looking at that particular research, right? And so part of this, we have a big problem right now with antibiotic resistance. And so some of you have heard of MRSA, right? Methicillin-resistant Staphylococcus aureus, but there's others, right? There's VRE, vancomycin-resistant Enterococcus. There's VRSA, vancomycin-resistant Staphylococcus aureus. There's MDR, TB, multi-drug-resistant tuberculosis in the United States. But in places like India, we have XDR TB, which is extremely drug-resistant tuberculosis, and that means that we have no antibiotics to treat. We're having to move into the old-fashioned way of treating patients because of things like CRE, right? Uh, Colicin-resistant Enterobacteriaceae. We no longer have effective antibiotics to treat some infections, and that's a problem for us in the States also, because in the United States, we have a lot of people who are immunocompromised, right, for lots of different reasons, including diabetes, right? And so we have a large population of Americans that are diabetic, right? And, I'm Hispanic, and so I can say this without people getting all crazy, but one of the biggest demographics that are affected by diabetes are the Hispanic population, right? And they are the very non-compliant, right? You tell them something and they don't follow your direction, right? You tell them, well, you shouldn't be eating cookies or whatever. And if you're like my mother, when she was alive, she'd hide cookies from me so that I didn't know she had them, but I knew she had them, right? And so that's a problem, right? Because we have all these diabetic patients, and these diabetic patients are more likely to have an infection that and have a negative outcome from having that infection. My mother's left leg was amputated because she developed an infection of the foot, and the only way to save her was to amputate, right? And so I'll tell you that story later in the semester. But it's an amazing thing to think about how many people are affected by diabetes, and really not only personally affected, but also as a familial unit, how many people are affected because they're going to have to be caregivers to these individuals who have end-stage renal disease or other problems like that because of diabetes, right? Good. Any questions about the problems that we have facing us with antibiotic resistant microbes? I myself had my own personal medical issue, right? So about 10 years ago, I had to have my shoulder replaced, my left shoulder. You can see I'm here in the hospital, St. David's, excellent hospital. Um, but it was an old football injury. It's just crazy that I played football in high school. I just can't imagine I did that. It's just not very smart. But um, I was in the hospital for 3 days, and so I woke up after surgery. I saw my wife, and I said, "Everything okay?" She said, "Yeah, somebody's here to see you." And I turned around, and there she was, Katie. This is my ex-student. And she said, "Hey, Pro V, do you remember me?" And I said, "Yeah. How you doing, Smiley?" Because that's what I called her, because she was forever smiling. And she read books all the time, and I love books. And so she and I talked about all these different kind of books. And while I was in the hospital, she changed her schedule so that she could be my nurse. And I was very grateful for that because she and I problem solved the whole time. I was having problem with Demerol. It wasn't allowing me to go to the bathroom. So she and I problem solved. We asked the attending physician um, to prescribe another um, pain medication. So I got Percocet. Well, then I was tacky because of that. And I said I was flushed, I was tacky. I was like, oh, this ain't working. So we decided on tramadol. And I was very, very, very happy that I had Katie there who would listen to me and then help to be my liaison between the attending, right? Because sometimes, the attending is just too busy to really pay attention to you, but the nurses always do, right? That's why I teach courses like this for nursing, because I want you all to be as successful as possible, right? So here you can see, remember I told you, the two things you should think about or you should know every day when you come into my classes: where do you stand in this course, right? Y'all have A's right now. And the second thing is, why is this important? My wife said that when I got out of surgery, the physician came in to check on me, and she said, "You grabbed him by his tie and pulled him to your face, and you said, 'When can I leave?'" And I said, "Oh, well, okay. I don't remember exactly doing that, but I know I would have done that. Why do you think I asked the physician, 'When can I leave?' What was my motivation for asking that? What do you think?"
Probably go teach some more.
What's that? I'm sorry, David. What was that?
Probably go teach some more.
Oh, okay. Well, Jenna, what did you think?
To like avoid having to stay longer so you don't get an infection or something.
That's exactly what I was thinking, right? I'm like, I want the hell out of here because, sick people go to hospitals, and so they leave all their cooties behind, and I didn't want any of those cooties. I'm like, "Phew, let me out of here." And the physician told me you have to stay at least 2 nights. I'm like, oh hell. So after that second night, I was calling them up and saying, "Hey, I'm ready to go." He said, "Well, I told you what I said. I've already made sure that my digestive system was working properly." I said, "I'm ready to go. Let me out." Uh, so he discharged me, but it, it took the people at the station at that particular unit like 4 hours to discharge me. I'm like, really? Just sign the papers and let me out. But it took them a little while to get me out. But I was ready and I was glad to get out of there. Right. Thank you for the surgery. I have about 85% use of my shoulder because of the surgery, but I don't want to stay in the hospital any longer than I have to because of nosocomial infections. What's nosocomial? Who can tell me what nosocomial is?
It's an infection that you gain when you're in like a hospital setting.
Very good, Miss Jenna. And we're trying to get away from using that term, although we'll probably always use it because it sounds good and it sounds very scientific, nosocomial. But we're trying to use the terminology HAI, right? HAI, hospital-acquired infection. And if you get an infection out, like you're a wrestler and you get a staph infection from the wrestling mat, that's out in the community, so we call those CAIs, community-acquired infections, right? So again, you can see that it's gonna hit home. Somebody in your family is gonna have to go to the hospitals. First time I ever been to the hospital, right? And I worked in hospitals before, but I was never in the hospital. So I wanted out as soon as I could. But that's not the only issue why a course like this is important. Another course is because we as humans are disruptors. We are disrupting the environment. And when we disrupt the environment, we cut down the forest, we destroy jungles, and all these infectious agents are in there, right? And we're releasing them, right? We believe that SARS, right? This particular magazine was from about 20 years ago when the first SARS epidemic, the first coronavirus epidemic hit. And that one didn't really affect us in the United States, but it affected a lot of Asia and places like that. Then there was another one, MERS, right? Middle Eastern Respiratory Syndrome. It didn't really come here. We had a few cases, but mostly it was in that region of the Middle East, Africa, and that part of the world that really affected. But SARS-CoV-2, the third version of the coronavirus, showed up and it's knocked us for a loop, right? And so this, we believe, as we look at the data, and I'll explain this to you later when we talk about viruses, is that simply we're having mutations of these viruses because of the close proximity to animals, and the animals in the jungle or the forest have these different viruses. It don't really affect them, but then they interact with the human, and then they become dangerous to the human. They mutate, and so we know that SARS-CoV-2, what you all will know as COVID-19, has a part of the virus that is pangolin in nature, part of the virus that is bat by nature, and part of the virus that is um, human by nature. And so when it first came out, people were talking about this scientifically. They were talking about the novel SARS-CoV-2 virus. The N before it means novel. That means we've never seen it before, right? Because of that, we didn't have any immunity, right? And so it's killed, I think, almost 1 million people in the world, which is not good. But if you think about this, there's a virus out there that's even worse than COVID-19. How many people have heard of H5N1? How many people have heard of H5N1?
I
Anybody ever heard of it?
I think I have.
Jenna, what do you know about it?
I want to say that I heard about it in a documentary or a movie, but
Oh, okay.
past that,
Yep,
I'm
yep.
not sure.
Look, that's okay. So I know you haven't heard of H5N1. That's a serological type of influenza virus. But I know that, yeah, it's flu. But I know you've heard of it this way, avian flu. How many people have heard of avian flu? Yeah. So H5N1 is avian flu. It has a 60% mortality rate, right? Think about that. 60% mortality rate. Yeah. So what that means is that if this hits, like I think it will, in my lifetime, I think we're going to be hit by H5N1. That means that it's so dangerous, it's so virulent that let's take 1 million Americans. If we have 1 million Americans and they all come down with H5N1, how many of those 1 million Americans are going to die? Correct. 600,000. Mhm. Now we have 320 million Americans. So multiply 600,000 by 320, and you get to see the ramifications that a viral infectious agent can have, right? Because it's easily spread. H5N1 is not easily spread just yet. It's a few mutations away from that, but it's going to get there eventually. And if you think we weren't ready for SARS-CoV-2, COVID-19, we're not going to be ready for H5N1, right? Has anybody watched the movie Contagion? Anybody watched the movie Contagion? So watch it, because even though, it's a movie, it really tells the story about how an infectious agent can get out of hand very quickly. Jessica, I want to address your question. How do mutations happen? It happens because RNA viruses that are enveloped, like influenza and like COVID-19, can mutate when they come in contact with other individuals and they go from one body to another, right? But they can also go to other animals, right? So right now, there are some studies that are being talked about in Austin where some of our dogs are getting COVID-19, but it's not affecting them as bad as it affects humans, right? But if the virus COVID-19 is in a dog and it gets a little bit of dog pieces, and then it comes back out and it becomes even more dangerous, right? Because now it has a pangolin part, it has a bat part, it has a human part, and a dog part. That's a mutated virus, right? So unfortunately, my dog passed away a year ago. So I haven't really thought about it too much, because every time I think about dogs being sick, I think about my dog. So I try not to think about it so much. But I, if the dog looks like they have respiratory distress, now that's okay. It's part of life. It's part of life. Things that you love die, right? And so um, that's okay. I appreciate it. But listen, so if your dog appears to have respiratory, you might take them to the vet. I don't know. I mean, when my dog had cancer, passed away because she had cancer, we spent over $1,000 on her, right? And to me and my wife, that was money well spent, because she was our kid. But some people might not be able to do that. So I don't know. There doesn't appear to be any mortality associated with it in dogs, at least for the studies I've read. So Sarah, just kind of watch your doggy, and if they start to not want to participate with you, because dogs are always going to want to participate in your life, right? They always want to go with you. They always want to do stuff. Yeah. Then maybe you want to take them to the vet, right? I myself have been exposed to COVID-19 probably 4 times. I don't think I've had it, but I don't see a need to be tested for it, because I'm not exposing anybody. I'm mostly staying home. I do go to HDB. I do go to Riverside, and when we go back in on campus, I'll be wearing a mask or a face shield when I'm talking to you in lab. And so I'm going to do my best to make sure that I don't expose anybody, right? So I haven't been tested for it, although I did order some of the home tests from the government. So when those come in and I feel like I might have symptoms, I might do my own testing at home, right? But I don't see a need to understand if I've had COVID or not. A lot of people go stand in line to get tested, but they're going to concerts or they're going to work or something. ACC will have testing on site, so if I feel like I got to do that, I can do that there also. So I just don't feel a need to go get tested. It's not that important to me to know if I've had it or not. The likelihood is that I've had it, but I don't know that for sure, because if I've had anything, it's very mild, right? I just I just don't see a need. But just think about this, right? This is Austin, Texas. Um, in Austin, Texas, we're kind of a city who likes to like be good to the environment. We like to do our own things. We like to have organic stuff. Are you with me? And so even my neighbors, like next door, they've got chickens. Those chickens come over here and they lay eggs in my yard, which is fine. But we have a person down the street that's got goats, and A little bit further down, a couple people got horses and things like that, right? So those are all close proximity with animals, right? And I don't know if you know this, but I think it was Israel. Now somebody can look it up and make sure that I'm telling the truth. But I think Israel killed, I don't know, 250,000 chickens because avian flu was breaking out in that chicken population. And so somebody check that for me. You can look it up on your computer. And if I'm wrong, say Provi, it wasn't that country, it was another. But I think they killed over 250,000 chickens because avian flu was breaking out. Now they did that for two reasons. Number one, it was causing a strain on the chickens because they weren't going to be able to sell those chickens. And number two, from a medical perspective, they didn't want avian flu in those chickens very long because if they're in there, they could mutate and then they might come out and cause problems in humans and other animals, right? And so it's really important that we understand that even though, you know, people want to have animals in their backyard, which I don't have a problem with, um, that we just need to be careful that those animals stay healthy. Okay, questions? All right. How do we know? I'm sorry, missed that, Jess. Go ahead, Jess.
So how do they know that they had it? Like, how do they test that?
What for? What COVID?
No, like for the avian flu when the chickens had it, how were they so sure they had that?
Oh! Because of the symptomology, then what they could do is they could test. They could just take a little bit of the sputum from the chickens, the mucus, and then they could look at that and real quickly, right? Nowadays we can do it within a day's time. We can because we have the genome for H1N1 plus a lot of other viruses, and we can run anything that's in that saliva or in that mucus, and we can run it against a known panel of different infectious agents, and therefore we know. So it doesn't take very long at all. And check this out. If you had TB in the 70s, it probably took 2 weeks for a confirmation test. If you have TB today, we have the technology in 4 hours, we can tell you if you're TB positive or not, right? So that's how far science has come in what, the 40 years since those since you could think about, people having TB in the 70s, right? Thank you. Thank you for checking on that. Was 250,000 correct or was it more? I don't. I think 250,000 was about the number. Amendera.
There's a couple websites, and it doesn't give like an exact number, but it said
Hmm.
like one website says over 500,000.
Yeah, I heard that number also, but I wasn't sure. That's why I went down to 250,000 because I never, I never want to be accused of lying to you.
Yeah,
Provi, you're throwing hyperbole at me, and so
yeah.
I'm not going to believe you, right?
A
So
lot of
I'm
these
always
say
going to use a
big
lower
numbers.
number.
Yeah, they all
Yeah.
say big numbers, so you're probably right.
All right. I read a lot, and sometimes when I read, I'm like, where did I get that information? So I got to be sure that I'm giving you the right stuff, right? I'm trying to keep you updated what's going on in the world because a lot of you do not watch the news, right? Can anybody in my audience right now in our class, can anybody tell me if they watch the news? How many of you watch the news? Oh, David does. So Alexi's like, I refuse to at time. Okay, I should, but okay. Alexi, why do we refuse to?
I feel like there's so much misinformation and everything
Oh, yeah,
was
yeah, yeah, yeah.
so
Okay.
it's just a lot and then you have to go back and research it yourself and I'd rather just hear it from the people at work that actually do the research.
Okay. I'm okay with that. I tend to watch David Muir and then the BBC, and then I correlate what those two things are saying, and those typically give me the correct answers that I'm looking for. But you're right. I have to go and do my own research, right? Because that's the problem with the people in the United States nowadays. If you say something enough times, it becomes people's reality, right? And that's what I'm trying to get you all to think about um, when you are looking at things is but most people will say I never watch the news probably because it's all bad news. And that's true also. They never do good things, right? The very end, David Muir like does something happy, right? But for the most part, it's all bad news, right? Oh, is that right? What? Oh, wow. Um, I I really want to. I mean, maybe you could give a little presentation on your capstone at some point, because I think that'd be really cool. That's a really cool topic. Okay, moving on. We're now going to start to transition about talking about how we classify things. And so we have to start with getting a little bit of background. So I know we've talked about prokaryotic and eukaryotic. Let's pick on the new guy, Eric. Can you tell me what the difference between a prokaryote and a eukaryote is?
Yeah,
Anybody? Okay, what is it, Eric? Tell me, what's
sorry,
a
I just had to
prokaryote?
turn on the mic. It's the nucleus is different.
Or it's lacking, right? There's no nucleus. So with
Yeah, yeah,
prokaryotes, no nucleus, right?
yeah. They just
No
have free
nucleus.
flowing DNA, and then eukaryote they have a nucleus in it.
Okay, I'm good. So if you think about from now on, I want you to think about prokaryotes as a bacteria, right? Now, many people don't care about the bacteria. They're not like Pro V. Pro V loves the bacteria because he understands their importance, right? Not only from a clinical perspective, but also from an environmental, ecological, everything perspective, right? And so many people don't really care about the bacteria. Can somebody tell me another type of bacteria that we've mentioned, but I said, we're not going to really talk about these guys too much because they don't really have any clinical relevance. Can anybody tell me another bacterium that we've talked about? Yes, Jenna,
The archaea.
the archaea, right? So other people have said it also, but the archaea, the archaea. So those things are prokaryotic. Everything else that is alive on the planet is eukaryotic, right? Everything else. So that includes all the things that we love, our dogs, our cats, our birds, koala bears, snakes, lizards, insects, flowers, trees, whatever. Everything else that is on this planet that is alive is eukaryotic, right? And so, of course, more people think about eukaryotic organisms all the time, unless you're like me or a complete nerd like me and really think about the bacteria and how important they are, right? And I'm going to show you how important these things are throughout this semester, okay? But really, it's about what is alive and what isn't alive. And so there are about nine different characteristics that things have to meet in order to be alive, right? And so Amandeta has had me for 1406, so we've talked about this in that course. We're not going to go so far here because that's a little bit outside the scope of the course, right? But what I will tell you is if they can't meet these two requirements, right, then they're not going to meet any of the others, right? But so the two requirements that I'm going to say are very important is that in order to be alive, things have to metabolize, right? And they have to reproduce, right? So metabolism means that they have to consume nutrients or make their own nutrients and then use those to make ATP and then use ATP to make other cellular structures. That's metabolism, right? That leads to reproduction, right? Because reproduction is about producing more organisms or producing more cells. Most people will think about reproduction because we're humans and that's the way we think about sexual reproduction, right? Woman and a man having a baby. But that is such a small part of all of the reproduction that occurs on this planet because most organisms will only reproduce asexually, right? And if they're bacteria, they undergo binary fission, right? Because they don't have a nucleus, they can't undergo mitosis. And even us as humans, we undergo two forms of reproduction. We reproduce asexually, all of our body cells, skin cells, tongue cells, liver cells, everything undergoes mitosis, make clones of all those cells, right? And then in our sex organs, right, females produce ova and males produce sperm. And they come together and they form a unique entity, right? We talked about this, right? Everybody's unique. Uh, but so in order to be alive, you have to meet those two requirements. If you can't meet those two requirements, you're not alive. Viruses do not get past the first one. Viruses do not metabolize. Therefore, viruses by definition are not alive. Now, viruses are really good at infecting and really good at replicating, but they do not metabolize. Therefore, they're not alive. Okay? And prions are the same way. Really good at replicating, they don't metabolize. Okay? So how do organisms acquire nutrients, right? So there's some different ways. Trophic means to feed. Autotrophic means that they produce their own nutrients, right? And so there's a misconception out there that basically people will tell you, because if they don't know science, you always know somebody doesn't know science because they'll tell you that plants gain their energy by photosynthesis. That is not true. Plants produce sugars through photosynthesis. Then they undergo cellular respiration to make ATP energy, right? But they're autotrophic, right? Trophic means to feed, auto means they produce their own nutrients. Heterotrophic, trophic means to feed, hetero means that these organisms have to consume some other organism, whether it's alive or it's dead, right? So think about this. If you eat pasta, right? How many people like pasta? Anybody like pasta? Who likes pasta?
Me.
Okay. If you eat pasta, what are you eating?
Too much.
Too much. You're eating plants, right? Grains, right? That come from plants, right? Yeah. And so it's pretty interesting to think about that because people say, well, no, I don't eat something else. I eat pasta. Well, pasta was at one point a living thing, right? We've just processed it. And so now it's pasta, right? But if you eat salads, you're eating plants, right? And so it's an amazing thing to think about. Underneath heterotroph, there are two subs. There are two subheadings, and one of them is saprophytic or saprobic. And these are organisms that consume things that are dead or decaying. Can anybody give me an example of something that is saprophytic or saprobic? A vulture. That's exactly correct. Yeah. A caracara, right? Or also the mushrooms. The mushrooms are consuming things at one point were alive, but mostly dead now. Does anybody know how many people like mushrooms in their pizza or mushrooms in their salad?
I love mushrooms.
Me too. Me too. I love them. Do you know how we grow them? They take some straw
Dirt?
or leaves or whatever. They mix it with poop, turkey poop, chicken poop, cow manure, or something like that. And then they put the spawn on top of it. And then they put them in kind of a cooler, dark place. Serious. Look it up. They put in a cool, dark place. And then every once in a while, they go and cut them. And then they sell them to you, right? You might get a little extra poop for no extra cost, right? Well, I mean, I shouldn't say that because most of the time all that poo is composted. So most of the things that are bad in that poo are not there anymore, right? Well, I mean, Ava, it's true. So don't pretend it happens because I want you to be knowledgeable, right? But think about this. Hey, anybody, my dog, when she was alive, we'd be for a walk, we'd go on a walk, and she had a really great sniffer, right? They have tremendous sense of smell. And so she knew when there was a little bit of a snacky poo coming up because she could smell it way before I could even see it. So we'd be walking around and all of a sudden she would look up at me and I'd look at her and say, "What's wrong, girl?" And she would look down at the ground and it'd be, a turd or something. And she would eat it. She would eat it up, right? Because to dogs, poo is nothing more than nutrients. Are you with me? Especially kitty poo, because kitty poo has a lot of proteins in it, right? And so they love that. Beagles are the worst, right? They are, man, they're wanderers, right? They will find the truffle, I'll call it a truffle, poo, and they'll eat it up, right? It's an amazing thing because animals just see poo or even vomit, right? So anybody ever seen a dog vomit and then eat their own vomit? Because they're not going to let that nutrient go to waste, right? That's nutrients to them. Yeah. And so they'll eat it, they'll lick it up and eat it, right? Because it's just nutrients to them. The other subheading underneath heterotroph is parasite, right? And so parasites, by definition, are going to be consuming live tissues or cells or whatever, right? From things that are alive. And so we're going to be talking about parasites today in the lab, right? So it has a good correlation, even though we haven't really talked about parasites in lecture too much, right? Does anybody know? So I have three images here, right? One of them is a mushroom right here, right? And that represents a saprophyte. One of them is the underneath of a underleaf, right? And you see this opening. That is called a stroma. And that's the way that leaves exchange gases, right? They take in CO2 and they release oxygen, right? And then this other thing, you can see a tiger's got a fresh kill with it killed a water buffalo, and so it's having lunch. But that is heterotroph, right? It's not really a parasite, right? Because the definition of parasite is that the organism has to live with its host, right? But this is a special type of mushroom, not edible. Does anybody know what kind of mushroom this is? Anybody ever seen one of these? It, it's about it's about time for them to start growing. They grow in my backyard. They're called a stinkhorn. They're called stinkhorns because they emit an odor like poo, right? So they trick the insects, the flies and the beetles. They think, oh boy, lunch, and they land there and they walk around the mushroom and does exactly what the mushroom wants it to do, picks up its spores, right? And then they fly away. No lunch, no lunch here. They fly away, but they have their spores on them. And they land someplace else and they drop off the spores. And now you have the dissemination of the spores of the mushroom. And so other colonies can grow places, right? It's an effective way to disseminate the spores of a mushroom, right? It's really cool. And so these things grow in my backyard. And if they do grow this spring, if we get a little rain, they'll be all over the place. And we're back on campus, I will pick a few of them and I'll bring them in to the lab so that you can see them, right? Because I just think people need to be looking out for things when they're walking around at parks or even in your neighborhood, because nature's everywhere and the microbes are everywhere. And ever see that movie? What was it? The Sixth Sense, where that little boy, kind of scary, he said, "I see dead people," right? Well, I see microbes everywhere because I know what to look for, right? Some people look at trees, some people look at roses. I am a true nerd. I look at microbes. Okay, so let's transition now because we know about prokaryotic and eukaryotic cells. We know about how they can acquire nutrients. And so let's transition to talking about um, how we put things in groups, right? How we name them, right? So nomenclature is the way we name things, right? Taxonomy is how we classify things. And identification is how we specifically say, this is the organism that is causing the problem, or this is whatever, right? because we have special ways of identifying things, right? And it all started with Carl Linnaeus, right? With Carolus Linnaeus, who basically said, remember we talked about this first day of lecture? We said, Linnaeus said everything was either a plant or an animal, right? And then from there, it just became this beautiful thing where we started to differentiate organisms based on characteristics, right? And so one of the first taxonomical schemes that we had is what we're seeing right here. And you can see that we started off with kingdom, then we had phyla, we had genus, class, we had order, family, genus, and species, right? And there's another part to the hierarchy. Does anybody know what it's at the very top of the hierarchy now? Does anybody know? What is the very top of the hierarchy? Anybody know? It's called a domain. And I'm going to tell you the story of how that happened. Okay, because it's important for people in science to know what the domains are. Can anybody tell me what the 3 domains of life are right now? I'll give 1 point extra credit to anybody in 5 seconds. 1001, 1002, 1003, 1004, 1000. You're about to learn something new.
I think
Okay?
I know it. I don't.
What is it, Alexi?
Is it eukarya, bacteria, and archaea?
Very good. That is correct. Very good. Very good. 1 point for everybody because several people were willing to take a chance, right? Everybody gets 1 point on the first practical. How about that? Nah, on the first exam. 1 point for everybody on the first exam just because you made me happy today. Very good. Oh, you looked it up, Cesar. Cesar, don't say things like that. Take the credit. Okay? Yeah. Questions. I'm going to tell you the story now how this happened, right? It all started with Linnaeus. Carolus Linnaeus was an observational scientist. He was looking at things. He said, you know what? Everything is either an animal or a plant. And he started this whole idea of putting things in groups. But that's not what he's most famous for. Almost nobody gives him credit for thinking about how things should be arranged. What he does get credit for is he was the individual that came up with the first way to name things. He came up with the binomial nomenclature system. We know that now today as a scientific name, and it consists of a genus and a species. And so if we take the human, the genus for humans is Homo, and the species is Sapiens. Oops, that's start over. If it's written, that's an O, it's underlined. If it's typed, it's italicized. Okay? So how many people have ever heard of E. coli? Okay, good. E. coli is a bacterium. It's a specific bacterium. Its genus is E, and its species is coli. But the E stands for something. Does anybody know what the E stands for? What does the E stand for? Anybody know? Long word. Escherichia. Very good. Very good, Miss Luna. Sorry, Miss Rosa. But the E should be capitalized, right? Because the rules of the binomial is that the genus is always capitalized, the species is lowercase. You were probably just trying to write it, Rosa, so that you could make sure that you got the credit, right? Yeah. Very good. So it started there, and then other people came in and started doing different things. Whittaker came in and said, because we have this ability to look at things microscopically, and we have other ways to look at the nutritional requirements and things like that. He said that they were 5 kingdoms, and then it got broken up again to 6, right? But you can see that his early idea was that things were either animals, they were in a plenty, they were plants, they were fungi, they were protista, or they were prokaryotes, right? Now the prokaryotes eventually got broken up into 2 groups, and the 2 groups they got broken up into were, of course, the bacteria, which we used to call the eubacteria, and then of course the archaea. So Whittaker's work, 5 kingdoms, then eventually it got negotiated so that there were 6, right? But if we think about this, right, it was because of Linnaeus's work to start off with, right? So he put it into this order, and then people started looking at different organisms and coming up with ways to classify them. And it even makes their way into the movies every once in a while, right? So I don't know if you guys anybody ever watched Jurassic Park, the first movie? Anybody ever watch that?
Of
Yeah.
course.
So Laura Dern's character is looking at this leaf, and she says, "Oh my God, you know, this leaf, based on its venation and its morphological characteristic, it belongs to this particular genus and species." And she said it. I'm like, "Oh, good. They're talking science, right?" But that's the way it was, right? It was all about morphology, and she was an expert in ancient extinct plants. But she now saw this, and then about that time, they see the first dinosaurs on this island that they were on, right? And so it's pretty cool to think about how this might have happened, right? But it all based on this, right? So the way that we can further classify things now is based on some different things, right? We can look at physical characteristics, we can look at biochemical activity, what they take in as a nutrient and what they produce as an end product metabolism. We can look at serological tests. We can look at phage typing, right? Which viruses will infect them. We can look at nucleic acids, but it's even gotten more complex than that. And we're going to talk about how that is in just a minute. So this is Whittaker's classification. Remember, I told you that it got changed a little bit so that there were 6 kingdoms, right? And so if you think about these 6 kingdoms, if you will, then you could see that it started out with prokaryote, protista, fungi, plantae, and animalia. And then we divided up the prokaryotes into 2 groups. And so if I draw a line right here, we have the animals, we have the plants, we have the fungi, we have the protozoans, and then we have the prokaryotes. We have the bacteria, and we have the archaea. Okay, so 6 kingdoms, right? And it's all based on the 3 M's, and I want you all to know that the 3 M's. The 3 M's are morphology, metabolism, and the molecular techniques. And the molecular techniques are really fatty acid profiling, protein differentiation, DNA fingerprinting, and lots of other things, right? So because of that, we can differentiate these groups even further, right? And so that's the way it was for a long time. That's the way I learned it when I was in high school, right? And all through college. But about my second year in graduate school, there was another guy who was challenging the status quo. And let's take a look at this image right now. So Jenna, what is the emphasis on in this image? Is it prokaryotes or eukaryotes? What's the emphasis on?
Eukaryotes?
Uh-huh. It is the eukaryotes. Why do you think that? Anybody? Why do you
Be-
think the early emphasis was on the eukaryotes?
Because they're like the stuff that you could visibly see.
It's the things we care about. It's the dogs, the cats, the whales, the porpoises, the squirrels, all the pretty things, the plants, all the roses, you know, all those things, right? But the bacteria, nobody really thought about them, right? It wasn't until about the 1970s that people really started to see the importance of the prokaryotes, of the bacteria, right? And so a lot of research happened. And around the 1980s, a guy showed up. I was late 80s. I was in undergraduate school, about to go to graduate school in the 1990s. And so um, a guy showed up. His name was Carl Weiss. And Carl Weiss went to one of the nerd conferences I would go to. And he said, you know, we've been talking about these kingdoms for a long time, but really I'm here to tell you that based on my work, and his work was on using ribosomal RNA. So his work on ribosomal RNA basically said that there were only three super kingdoms. And he said the three super kingdoms are the eubacteria, the archaeobacteria, and the eukarya, right? And so if we think about it, this was one of his first ideas right here. Break them up into three different groups. And so people like at that particular conference, they kind of basically showed him very little respect, right? They didn't really agree with him. But about 3 years later, as I was leaving graduate school, Fox started to talk about, or at least they've been talking about it for a while, but it started to be known. Fox basically was saying, what Carl Weiss has been doing, I can attest that is true because I've been doing work with ribosomal RNA, my life's work, and I'm going to tell you that there are only three super kingdoms. So a lot of people started doing research, and a bunch of people jumped on board and said, we agree, right? And so they said, well, we need to incorporate this idea because now we've proved that there are really only these three super groups, but we don't want to call them super kingdoms because we already have kingdoms. So let's call them something different. And of course, they call them domains, right? And so that's how everything started. So then you have your three domains, and then you have your kingdoms underneath those, right? So you have domains, you have kingdoms, you have phyla, you have class, you have order, family, genus, and species, right? The three domains are the three domains are the bacteria, the archaea, which I'm going to just say right here, and then the eukarya. And underneath the eukarya are four kingdoms, and they are the protista, the fungi, the plants, and let's move this one up here, the animals. Okay, I'll write them in the chat for you real quick.
So, is there no kingdoms under the bacteria and archaea because it's
Nice.
already their own domain?
Correctamundo, that's correct. Sorry, I misspelled protista. Let me spell it correctly. Protista, the fungi, plants. Oops. Sorry, I'm having a little bit of difficulty with my eyes right now. Some kind of distortion. Here they are. But that's right. So, there, I think, said that there's no kingdoms underneath the bacteria and the archaea because they're domains. Now, there's a lot of work going on with, what should be kingdom and what shouldn't. I mean, one person has uh, proposed 18 kingdoms just under the archaea. All right, I'm sorry, 18 kingdoms just under the protista. Uh, I don't know if that's going to happen, but for right now, this is what's accepted. Right? There are other kingdoms underneath the archaea and some of the bacteria, but nobody said, yeah, this is the way we should do things. So right now, it's going to stay like this: 3 domains, and underneath the eukarya, 4 kingdoms. Okay, you need to know those. Okay, questions. So let's talk about the scientific name, right? So the rules of scientific name are that the genus is the first name and it's always capitalized. The species is always the second name of the scientific name, and it's always lowercase, right? If you type them, they need to be italicized, but if you handwrite them, they're underlined, right? And the other rule of the binomial nomenclature system is the genus can be abbreviated by the first letter in the genus followed by a period, right? So therefore, Staphylococcus aureus, that's written here, Staphylococcus is the uh, genus, and aureus is the species, but it can be represented like this, okay? Where the S stands for Staphylococcus. So how did Staphylococcus get its name? So coccus is a shape, Staphylo is a arrangement. So these are cocci that are in clusters, and aureus on the periodic table, what is represented by AU? Does anybody know? Gold. That's right, David. So aureus is a derivation meaning gold. And if you look at the colonies growing on this plate right here, you can see they're golden. So this name translates into a coccus is found in clusters whose colony is gold. That's what it means, right? And that's how it got its name. Good. Here's another one, right? This is Escherichia coli, right? Escherichia was derived from Dr. Escherick, who was a physician who first characterized E. coli. And E. coli was actually recovered from feces of a mammal. And so, therefore, we gave it the name Escherichia to pay honor to Dr. Escherick, and then coli because it was first isolated from the colon of mammals, right? Any questions? Any questions? Okay, so Staphylococcus aureus, if you think about it, coming back to it, it's important that because it can cause all kinds of infections. As a matter of fact, the 2 organisms that I respect the most, and I've worked with a bunch of bad boys, is Staphylococcus aureus, and the other one is Streptococcus pyogenes. Yes. Let's see, Jenna, what's up?
I have kind of like it's kind of a random question, but the kingdoms are for all like living organisms. So do viruses like fall into that anywhere since they're
No,
not?
we
Okay.
don't have any. We don't have any kingdoms for viruses or prions because they're not alive.
Okay.
There is classifications for them, but they're not part of the kingdoms. Now, it's very interesting that you say that, Jenna, because there are some scientists who think that we should open up the classification system to adding the viruses because they're so important, but they're not alive. So there's a lot of resistance. Okay. So getting back, Jen, I hope I answered your question. Getting back to Staphylococcus aureus and Streptococcus pyogenes, which I wrote in the chat. Streptococcus pyogenes is really important because it causes strep throat, but it also causes things like necrotizing fasciitis. It causes things like pimples and acne. It causes things like vagina. It causes all kinds of different things, meningitis, all kinds of different things, right? But the species name Pyogenes, genes means the former of pyo, pus. So this is a pus former, right? And that's how it got its name. So Streptococcus, coccus, round things that are strepto in chains that form pus. That's what its name means, right? And so that's really interesting if you think about how these organisms first got their name. Okay, questions? All right. So, yes, Jenna? Jenna?
It's Jess. I'm sorry.
Oh, sorry, Jess. What's up? My bad.
So it's okay. My question is, I'm not sure if I misunderstood you. You said that staph and strep can turn into other things.
One
Is
more
that what you
time.
said?
Staff what now?
I'm not sure if I heard you right, but did you say that strep or the staph could turn into other things? Is that what you said?
No, I never said that.
Okay.
So what I said was Staphylococcus aureus and Streptococcus pyogenes are the two organisms that I respect the most because they can infect every single cell of the body.
Okay. Thank you.
Okay. Mm-hmm. So as we start to end this conversation about classification and phylogeny is the study of how everything on the earth is related to each other, right? Evolution explains life once it's here on the planet. Evolution never explains how life started. That was never part of it. People who don't know any better will make all kinds of, as you guys say, uh, false facts about evolution. Evolution only explains life when it's here on the planet, never about where life started. The two preconceptions of evolution are that if there's a new species, that new species came from a pre-existing species, and that pre-existing species mutated and now is a new species. Okay. The second thing that evolution states is that organisms that have characteristics that are similar share those characteristics because they have a common ancestor. And so I showed you I showed you the kingdoms, the tree of the kingdoms that Whittaker proposed. I'm going to show you now the most important, one of the most important things that have been published in the last 15 years, and that's the current phylogenetic tree about how everything on this planet is alive. So, Jenna, do you remember what we said about what the emphasis was with the Whittaker classifications? Was it prokaryotic or eukaryotic?
Eukaryotic.
Mm-hmm. Right. So in 50 years, this is what we've come up with now. This is the most recent version of the phylogenetic tree. Ladies and gentlemen, everything that I am now circling is the bacteria. Everything that I am now circling is the archaea. And now what I am designating by the circle are the eukaryotic organisms. And Oponsacantha is right here. That's the fungi. And we are about right here as humans, right? Let me make that a different color so you can see it. We are about right here. So now, what is the emphasis? Is it prokaryotes or eukaryotes? Prokaryotes. And what that means is that the prokaryotes have been around a long time, right? But the eukaryotes are the new kids on the block. We've not been on this planet very long, right? But the bacteria have been. They've been here for a long time, right? And not only that, the other thing that blows people's mind is the common ancestor we have with the nearest other domain is right here. And so what do we as humans what are we most related to? If we think about domains, the archaea, right? So some religious folks will tell you evolution says that we evolved from apes. That was never true. We have a common ancestor with apes, but we never evolved from them. But if we told those people, you know what, we evolved from archaea, they might have a heart attack right then and there, right? But that's true, right? So in about the span of the last 80 years of science and scientific discovery, we've learned a lot about the relationship of organisms on this planet because we share things, we share DNA with everything that's on this planet, right? And it's an amazing thing to think about. But yet, people are stuck with, well, science says we evolved from apes. Science never said that. People who don't understand science said that. Okay? Any questions? All right. Time to go talk about tools. So we're about to begin talking about cells, and we're going to talk about bacteria, and we're going to start that on Thursday. But today we are going to cover tools, right? The tools that we need for our lab to be able to distinguish the organisms that we're going to be studying, right? And so we're going to start with the bacteria, right? If you think about the bacteria, yeah, they're real simple in design, but look at the different parts of the cells, right, that they have. So they have these projectiles that have come out their hair-like structures are called fimbriae. They use those for attachment. They have inclusions. They don't have organelles. They do not have organelles, but they have vesicles and vacuoles that they hold things in, right? Vacuoles. They have cell walls. We'll talk about that. They have a plasma membrane. We're going to talk about that. Outer membrane is only found in gram-negative, and we'll talk about that. They have endospores. Only gram-positive organisms produce endospores. So we're going to be talking about that. It's a survival mechanism. We'll be talking about the fluid matrix within a cell, the cytoplasm. We'll be looking at flagella and why they're important. We're not going to talk about active cytoskeleton. We'll be looking at ribosomes and why they're important. And in bacteria, they're free floating in the cytoplasm, but in eukaryotes, they're attached to the rough endoplasmic reticulum. We'll look at pili. A pilus is a gram-negative mechanism, and we're going to look at that. They have plasmids. We'll look at those things. We'll look at the nucleoid or the DNA. It's one chromosome only of the bacteria, and then we'll look at the glycocalyces, the covering of the cell wall, where some bacteria have them and some do not. Not yet. We'll talk about it in detail later. Okay, not yet. Okay, I'm telling the story. As you know, because you've had me before, I like to build on what we currently know, right? And so I'm telling the story. So the three tools we're going to discuss today are morphology. What does it look like, right? What's its shape? Microscopy, how do we look at them? And then staining, right? How do we elucidate what they really look like? Because a lot of them are colorless and we can't really see them, right? And so those are the tools that you'll need to know. And let's talk about them, right? So morphology, we've already talked about the fact that bacteria, we're going to talk about bacteria first. Bacteria have three distinct morphologies. There are cocci that are round. There are bacilli that are like rods. And there are sporili. And the spirilla, spirillum is singular, spirilla is plural, but they can look like an S. They can look like those birds that you once drew when you were in elementary school, or they can be really tightly helically coiled, right? So there's some variations in the bacillus. They can be a coccobacillus, which just simply means that they're a plump little rod-looking thing, right? They can be vibrio, and vibrio are comma-shaped. May look like that, right? Or they can be pleomorphic, right? Pleomorphic just simply means that if you look at them under microscope, they have a lot of different shapes, and you're not sure, so you say it's a pleomorphic organism. Okay, so you should know those different shapes, and we're about to be able to use them because I'm going to quiz you in a little bit about these different shapes. Okay, good. Any questions so far? Okay, so here is um, an image showing the three morphological distinctions of bacteria, right? Cocci, right? Rods or bacilli, and the spirilla. What do you notice about the cocci? Are they perfectly round? Are the cocci perfectly? No. As a matter of fact, when you guys are going to find out, when we stain the bacteria, sometimes they become distorted because of the interaction between the dye and the cell wall. And so there can be distortion that occurs because of the interaction between the cell walls of bacteria and the dyes that we're using. And especially for the cocci, it makes them look kind of funny, right? And so we need to be able to understand that. Okay, so if we then go on and look at arrangement, right? So we're looking at morphology still. How do they look? We talked about cocci being round and spherical, right? But now they have different arrangements. So they can be found um, by themselves, right? Single, like this. They can be found in pairs, which we call diplococci. They can be found in tetrads, which are found in fours, right? They can be formed in an irregular cluster, right? And if they're found in a regular cluster, then the term staphyl is put in front of them because staphyl is Greek meaning clusters like grapes, right? Or they can be found in chains, right? And so if they're found in chains, we put the prefix in front of coccus, streptococcus, right? And that means that these are cocci chains, right? I'm not going to even talk about Sarcina because we almost never use it anymore except for old school people. And so I need to take that out of this presentation completely. But those are the things. Now, here's the importance of knowing these things, right? So if Dr. Almendida sees me in the hallway and says, Provi, this particular specimen was taken from an 80-year-old spinal fluid, right? And she's complaining of having stiff neck. She's got high fever. Will you go take a look at this and tell me what you find out? I just need a gram stain real quick. And so I go back and I go to the lab and I find a gram negative diplococci that was found in CSF. Right? I go back to Doc and I say, Doc, I found a gram negative cocci in her CNF. And so she says, well, what do you think based on her symptomology? I say she has got meningitis, right? She's got Neisseria, meningitis, which is not a bug you want to mess around with, okay? So we need to start her on empirical therapy right. Not only that, but this thing is so dangerous. It's got an 80% mortality. Anybody who came in contact with that patient needs to be treated prophylactically with antibiotics against Neisseria meningitidis. Okay. What does that mean? Treated prophylactically. What does that mean? What does that mean? No clue. Just in case. I like that, Sarah. Just in case. Very good. Okay. So now I'm walking down the hall, microbiologist of the hospital, and I come in contact with Dr. Sarah. Dr. Sarah says, "Hey, Proveen." Oh, I'm sorry. "Hey, Felix. Hey, I found this. This is a specimen of an exudate that came out of the head of an 18-year-old penis, right?" And so I'm like, "Okay, will you go and check it out and tell me what you see?" And I come back and I say, "Doc, I found this in the exudate. Got a lot of neutrophils in it, but I have a gram-negative diplococci, right?" She says, "What do you think?" I said, "Well, the your patient has got gonorrhea, right? Neisseria gonorrhea, right? And so you need to start them on empirical therapy for gonorrhea, right?" So again, understanding what the symptoms are telling us, understanding the place that the specimen was taken from, and understanding a little bit about the patient, we can figure out very quickly how we can help them, right? And that's just by using morphology and arrangement and staining as a tool, right? So this is why that's important, right? Because you guys are going to hear this all the time. "Oh, the patient's got a gram-positive diplococci," right? And so you're going to hear these all the time. And so we want you, right? We want you to understand this stuff, right? The bacilli are less varied. They're not as they're not important as diagnostically, right? So we can find those organisms sometimes in single, right? We can find them this. Sometimes we can find them in pairs and they become a diplobacilli. Sometimes we can find them in chains and they become a streptobacilli. And sometimes they can arrange in palisads, right? Side by side, and they're attracted to each other. So they tend to cluster up like this. And so when they're like this, we say that this is cording. And if it's from a bronch lavage or the lungs, I'm pretty sure the person has tuberculosis because this is the way TB acts. Right? Again, using just morphology and arrangement and what do we know about the patient to make a very early diagnosis. But we start treating, and if we're correct, we're 2 days ahead of the ball game, right? Whereas if we waited, the patient's going to get sicker and sicker, and now we're behind, right? We're behind helping the patient. Okay, so can you see how this is important for us to understand? Okay, questions? Yes, Jess?
So I have a sorry. My question is, I don't know if it sounds a little dumb, but you said
No
that
such thing.
if you were to find it in the lungs, it'd be considered TB. So could it be found like these types of bacteria, could it be found in different areas?
Yep, because tuberculosis is not just an infection of the lungs anymore. We find TB everywhere in the body nowadays. So
Okay.
yeah, and there are other forms of mycobacteria that are found in other parts of the body, right?
Okay,
Yeah.
that makes sense.
Okay.
Thank you.
Anybody else have questions? Okay, let's move on. Second tool is a microscope. So simple microscope is nothing more than a magnifying glass, right? And so we can use that to look at structures that are a little bit bigger, right? A compound microscope is the one that we'll use all the time. It has two different lenses and we can see almost all the microorganisms except we cannot see the viruses or the prions because they're too small, right? But we can look at and I'm not going to talk about the microscopes very in great detail because we've already covered it in lab. But just know that what a compound scope is and that it has two lenses. It has the eyepiece that's got a 10 times magnification and then the objectives, right, that have different magnifications. Right? But that's the one we use most of the time. The other type of microscope that we can use, although we don't have one at ACC, they're way expensive, is an electron microscope. And there are two types of electron microscopes. There is an SEM, right? An SEM stands for scanning electron microscope. And these can magnify things 1,000 times to about 10,000 times the size of things you're looking at, right? So the semiconductor industry uses SEMs to take a look at their chips and their wafers to make sure there's no cracks or fissures in them, because then that's going to make them not work properly in our electronic devices, right? Biologically, we use them to look at surfaces of cells or the curvatures of the topography of the cells, right? And so they're important tools there. The other tool, the other type of microscope is called a TEM. A TEM stands for transmission electron microscopes. And this can magnify things up to about 100,000 times, at least the ones I have used. Now, I hear that UT, I've not seen it, and they probably won't let me in, but I hear that UT has a really great microscope that can magnify things almost 1,000,000 times the size that they're looking at, right? And that's what they're doing right now to look at the spike proteins of the coronavirus. I don't know if you know this, but most of the work that was done on the spike proteins, and the reason we have vaccines, was because of the work that was done right here at UT by a couple of researchers there, right? And that's pretty cool to think about, right? So let's look at how these microscopes can be utilized, right? So again, if you think about them, we're looking at the contours or the surfaces with scanning electron microscopes. And so here you can see is a bacterium, and it is on a filter. These little holes are the nominal pore size of the filter, and the nominal pore size of that filter is 0.2 microns, and that's a sterilizing filter for bacteria, in that it's going to hold back all the bacteria. Now, it's not going to hold back viruses or prions because they're just too small, they'll go right through. But the sterilizing filter can hold back all bacteria, right? And so I have a life straw. And so if I go hiking and I'm really thirsty, I'll stick my life straw in a puddle of water and I'll drink from it. And that's going to remove all of the bacteria that might cause me a problem. Not going to remove the viruses. But in nature, I'm not so worried about viruses out there unless there's a lot of animals around, right? So I'll drink from that, what most people wouldn't drink from, right? Because I understand, okay? But here's a paramecia, right? So a lot of times when we take these electron micrographs, they are grayscaled, and we can enhance their color by adding a little bit of color with the computer, right? And so here is an alga. You can see how beautiful the algae are. Those geometrical designs are only found in nature. Mother Nature is creative. Here is a reproductive organ of a fly, right? Here is some different algae. Here's a fungus that we talked about last time, right? Here's the vesicle, the phyllides, and looking at the spores. Here is an ova with several sperm attached to it. None of them, well, this one looks like it penetrated here. Here's the ova with the chorionic membrane, right? These are parts of the lungs. Here's a white blood cell attached to some things that don't belong in the body. And here are red blood cells. Here is a fibrin network of the red blood cells. So that's called a net. Yes, our bodies can trap infectious agents in the fibrin network, and then they're easy to phagocytize by our neutrophils and other things, right? This is a taste bud on the tongue. You'll never think about your tongue the same way. And then this is a biofilm containing thousands of different species of microorganisms, and they're all communicating with each other, right? For those of you who had anatomy and physiology, you know about cell communication, and Amandita knows because we talked about cell communication in 1406. But they don't have the mechanisms that our cells have, but they have other ways of communicating, right? Good. So a transmission electron microscope is huge. It takes the whole room, and you'd have to be on the bottom floor where there's no vibration and there's no noise because it's very sensitive. But now you can look at viruses and you can look at inside the cell, right? So here you can see is the influenza virus right here. This is a eukaryotic cell. There's a nucleus right there, right? This is influenza. This is Ebola. This is a chloroplast, and inside you can see the grana and the thylakoids. And then this is a rabies virus, and this is one of the most famous electron micrographs ever published. It was grayscaled. The people who published it added color to it to make it more visible and more attractive, so people would buy the magazine. This showed up on Time magazine in the early 80s. Does anybody know what this is? The green structures are the first ever images. That's right, first ever images of HIV. This was taken by the Pasteur Institute in France because they were the first scientists to take a picture of HIV. Good. Any questions? I'm going to take just a little bit more time and finish, and then we'll go to lab. So staining, right? So I want you to write this down. The cell walls of bacteria are acidic by nature. So write that down. Important. All right, I put it in the chat for you, right? Cell walls of bacteria are acidic by nature. What does that mean? Amandetta, what does that mean? Anybody can help.
I'm not exactly sure.
Okay. They have a low pH, Cesar. Very good. They have a low pH, right? So think about that for a minute, right? They have a low pH. So here are some of the very basic types of staining methodologies we can use, right? So we're looking at positive and negative staining. Positive staining sometimes is referred to as a simple stain. Right? So positive staining employs a dye that is basic. And if it's basic, it's going to be accepted by the acidic cell wall. There's going to be a chemical reaction at the surface of the cell wall, and the cell wall is going to be stained so that you get to see the organisms that are colored and you can really tell their morphology, okay? Although there might be some distortion, okay? If we look at the opposite one, right? The opposite one is a negative stain, and negative stains employ an acidic dye. And if we think about this, the acidic cell wall is going to repel, it's going to reject the acidic dye. And so you're going to, the stain is going to stain the background, but you're going to see the morphology of the cells as a silhouette against that dark background. There is no chemical reaction at the surface of the cell, right? So you're seeing the true morphology. There will never be distortion, okay, because there's no reaction between the acidic cell and the acidic cell wall. It's the acidic dye and the acidic cell wall, okay? Any questions? Okay,
So,
so
oh, sorry, I have a question.
go ahead, Ava.
So, are the different stainings like you use one on gram positive and one on gram?
Nope, not yet. Don't
Okay.
go there yet. I'm
Okay.
not there yet. Okay, hold off 1 minute.
Okay.
This is just these. That's a beautiful thought. But these two types of stain, right? Positive, simple, and negative are only used to differentiate morphology. and that's it. Okay? That's all you can tell from them. So if we talk about simple stains, right, we can use basic dyes, and those basic dyes, they can be things like methylene blue, right, which is a blue dye. They can be things like crystal violet, that's purple. There can be basic fusion, which is hot pink, or they can be safranin, which is red. But when you use these, the cells are going to be stained. Are you with me? But all you can tell from them is the shape and the arrangement, right? So what can you tell me about the shape and the arrangement of these cells right here? What can you tell me? What's the shape?
It
They're
looks like
coxy, and
cluster.
what's the and clusters. So these are coxy and clusters. That's all you can tell me. Now this one, what can you tell me about the shape and arrangement here? They're blue, right? These are purple. These are
Chain.
blue. They're chains, so they're coxy and chains. That's all you can tell me. That's it. Okay, good. But you can call these streptococcus because of the arrangement, and you can call these staphylococcus because of the arrangement. Okay. If we then look at a negative stain, again, using an acidic dye, here are two examples. The background is stained, and then you can see the silhouette, right? What's the morphology here? What's the morphology here? Don't be shy. Spirelli, very good. What's the morphology here?
Bacilli.
Bacilli. And what's the arrangement of the bacilli? Bacillian chains, that's right. So they can be called streptobacilli, right? You can see you're putting it together. You learn a little bit about arrangement morphology. Now you're learning about how we stain them, right? And I think it was Ava. Uh, I'm not sure, but I don't remember exactly. But she wanted to designate them gram positive, gram negative already, right? Uh, but we weren't ready for that. Now we're ready, right? We're going to move on, and we're going to talk about differential stains. Now, differential stains employ more than one type of staining reagent, right? And therefore, you can tell the differences between different cell types, or you can tell the differences between parts within the same cell, okay? And the most famous of them is the gram stain. So the gram stain uses two dyes, and they're both basic. One of them's purple, crystal violet, and the other one is red, safranin. Okay? And so we always talk about these with the gram reaction first. Now, we don't say what's the gram reaction, we say what's the gram specificity, right? And your answer to that is either going to be gram positive, gram negative. And then you're going to say the morphology after that. So we can have a gram positive coccus, GPC. We can have a gram positive bacilli or a gram positive rod, GPB or GPR. We can have a gram negative coccus. We can have a gram negative bacilli or a gram negative rod. And we can have a gram negative spirelli. Okay. What's missing?
Gram positive, Spirelli.
Uh-huh. They do not exist. There's no such thing. And the reason is because of the differences in cell wall composition. The cell walls of gram-negative cells are flexible, therefore they can bend. But the cell walls of gram-positive cells are rigid and they can never bend. So you can never have a gram-positive spirelli. So on a practical, if I showed you a purple spirelli and I asked for the gram specificity, you're going to say what? It's gram-negative. That's right. By definition, we can never have a gram-positive spirelli. And we'll talk about that on Thursday. So gram-positive bacteria stain purple, gram-negative stain red. And so if we look here, tell me what the gram specificity and the morphology is for this organism right here. What is the gram specificity morphology for this organism right here? You know, go ahead, say it. You can say it, go ahead. That's right, it's a gram-positive coccus. What's the gram specificity morphology of this organism right here? Go ahead. Gram-negative bacilli. Good. Or GNR, right? Now here's a test. What's here? What is here? What is this thing? Somebody tell me. Who can tell me? Be brave. Come on, you budding microbiologists, be brave. Tell me. GNC, but it's purple. GPC, but those aren't really cocci. Look very closely. What do you see? What are these elongated structures here? That's a yeast. Look, they're budding, right? So this is not a bacterium. This is a yeast. So the person who did this stain thought that they had a bacterium, but when they looked at it closer, they stained it, it showed them that it was a yeast, right? This is the power of staining, because when you look at them under a microscope without stain, they look clear. You can't tell, but you stain them, now you can really tell. Good. Anybody have questions? Very good
Yeah.
job.
So how do you know which one to stain? Like, can you stain both of them?
If it's growing on as a colony and you're not sure, you should always stain it. Always stain it.
With what?
Because the worst thing that can happen, Cesar, is you come up with an image like the one that's on the right hand side, right? And then you say, oh, these aren't bacteria, they're yeast. And then you can go do something else. You can go figure out how we're going to identify it. Okay. Well, did I answer your question, or was it were you
I mean, like,
were you
is it
go ahead.
one dye or is it two different dyes for the
No,
negative
two
and
different
positive?
dyes. Two
So you
different
stain them
dyes.
with both?
Both. And I'm going to explain that to you on Thursday.
Okay.
Okay.
Oh, because one of them will reject it and the other one won't, right?
No, they're both basic. Hold that thought,
Okay.
Cesar, for next time. Okay, I'm gonna finish up real quick, and then we're gonna take a break. But here, can everybody see what's the gram specificity? What's the gram specificity morphology and arrangement here? Just say it. You know it. You know what it is. Tell me. Gram-positive cocci. And what's the arrangement? Diplococci. Diplococci. Very good. What about these guys right here? What about these other guys down here? What about these guys right here? You know, gram-negative cocci. Very good. The one thing that's common to both of these are the neutrophils. And you don't need to know that just yet, but neutrophils always show up when there is a bacterial infection. Okay. All right. So acid-fast staining is what we use as a screening for tuberculosis. Do you remember what I said about the bacillus that or tuberculosis? How do they arrange themselves? Do you remember how they arrange themselves? They're attracted to each other, so they form cords, right? And so, unfortunately for this patient has their hot pink, right, rods, but you can see how they're all attracted to each other. This person has a very, very, very bad infection at TB. Right? Again, a tool that we can use to help to start to treat our patients, right? We might do some other tests to confirm that what we're doing is correct, but at least we can start treatment right away. And then the last thing, parts of cells and endospore stain. Endospores are produced by gram-positive rods only, and they're survival mechanisms, right? Unfortunately, they can cause a lot of problems in humans and other things. But here you can see the structures that are stained red are the parent cells, and the structures that are stained green are the endospores. So if I was to draw this for you all, remember I'm not that great a drawer, but I would draw a rod and it would be all red. And then the endospore would be green. Eventually what happens is the parent cell dies and the endospore is released. And the endospore can remain viable in the environment for years, hundreds of years, right? And so we're going to be looking specifically at endospores and why they're important. Okay? Anybody have any questions about anything we covered today?
I have a question.
Okay.